Hello everyone, my name is Yushan Li and I am a master student from Foshan University. Today I would like to share my study on how social hierarchy is linked to meat quality and flavor in chickens, with a focus on sex differences and the gut-brain axis. We all know that meat quality is one of the most important traits in chicken production, but even when genetics, nutrition, and management are carefully controlled, we still see differences within the same flock. So other biological and social factors may also need to be considered. Beyond traditional factors, behavior also matter. These differences are often stable, and they tend to be linked to stress sensitivity and physiological regulation. In our previous work, we found that behavior-related traits were associated with exploration, learning, quality, and gut microbiome pathways. So behavior may help explain why meat quality still varies within the same flock. Building on the behavioral variation we discussed earlier, an important transition occurs once chickens are kept in groups. In group housing, these stable individual behavior differences do not remain isolated. Instead, they become amplified through daily social interactions. These interactions are expressed as competition for resources, differences in space, occupation, and frequent aggressive or submissive encounters. Over time, through such repeated competition and interactions, a clear and stable social hierarchy naturally forms within the same flock. In fact, chickens are one of the classic animal models in which pecking order was first described. Once hierarchy forms, birds no longer experience the group environment equally. Rank affects both resources access and repeated social challenge. Over time, this may contribute to differences in behavior, physiology, and eventually meat traits. One possible rule is stress-related regulation. Chronic social stress can affect energy allocation and muscle physiology, but this is only part of the story. Another important rule is the gut-brain axis. Social stress can alter the composition and function of the gut microbiota, leading changes leading changes in metabolites such as short-chain fatty acids. These signals can in turn influence brain function and neurotransmitter balance. Importantly, some studies also suggest that the gut microbiota itself may play a role in social rank regulation. So the gut-brain axis may help link social hierarchy to changes in behavior, neuron chemistry, and eventually meat quality. Based on this background, we ask two main questions. First, does social hierarchy contribute to variation in meat quality and flavor within the same flock? And second, are these differences linked to microbiota neurotransmitter pathways? And do they differ between males and females? To answer these questions, we use 72 Qingyuan partridge chickens, including 36 roosters and 36 hens, housed in 12 same-sex groups. Social hierarchy was assessed by repeated feeding competition tests using a feeder that allowed only one bird to access feed at a time. We then performed behavioral tests. At the end of the trial, the birds were humanly slaughtered, and we collected muscle, fecal contents, and hypothalamus for downstream analysis. We quantify social rank using Davis score based on the frequency and direction of competitive interactions during feeding tests. As you can see, high, middle, low rank birds remain clearly separated over time in both sexes, and the final day score was highly correlated with the 7-day average, confirming that the ranking was stable and reliable. We first examine behavioral patterns across social rank. In roosters, middle rank birds show greater exploration, whereas high rank roosters show a more vigilance-related profile and also had higher body weight, higher feed intake, and lower FCR. In hens, middle-rank birds show more locomotion and maintenance behavior together with lower vigilance, and they also show the lowest FCR. High-rank hens had higher feed intake and feeder visits, whereas low-rank hens show the highest FCR. So rank was linked not only to behavior, but also to feeding strategy and efficiency in a sex-specific way. We then examined whether social rank influenced meat quality and flavor. In roosters, middle-rank birds show higher breast yellowness and greater fine tenderness, while high-rank roosters show the most favorable overall flavor profile. The greater fine tenderness in middle-rank roosters may be partly consistent with their more exploratory behavioral profile observed earlier. In hens, high-rank birds had brighter breast color, whereas middle-rank hens showed the most favorable flavor characteristics. Low-rank birds in both sexes also tended to show stronger responses in sensor channels associated with less favorable volatile patterns like W1C and W3C. Overall, social rank was associated with both meat quality and flavor variation. To understand these rank-related differences, we next look at the gut-brain related signals in roosters. High-rank roosters were enriched in ruminococcus, which suggests a more favorable microbiome metabolic state. They also show higher cycle isobutyrate, which is consistent with more favorable flavor precursor metabolism and also fits the better flavor profile we observed earlier. In contrast, low-rank roosters show higher gut levodopa and higher hypothalamic adrenaline. These two markers together suggest stronger activation of stress-related catecholamine signaling. So in roosters, the better profile appeared to align with a more favorable microbiome metabolic state at high rank, whereas the poorer profile aligned more closely with stronger stress-related signaling at low rank. And in hens, the pattern was different. Middle rank hens were enriched in parabacteroids. This is important because parabacteroids is linked to bile acid and lipid metabolism, which may support more favorable flavor-related metabolism. Middle rank can also show higher propionate butyrate, suggesting a more active related microbiome state. In addition, middle rank hens had higher cycloserotonin, whereas low rank hens show higher hypothalamic kynurenine because serotonin and kynurenine are both downstream products of tryptophan metabolism. This suggests that the middle rank hens may have been in a more balanced metabolic state, while low rank hens were more shifted toward the kynurenine pathway. This also fits with the earlier results, where middle rank hens show a more stable behavioral profile and a better flavor profile. Taken together, our results suggest that social hierarchy was linked to meat quality variation through clearly sex-specific pathways. In roosters, the better profile aligned with more stable resources access, lower stress-related change, and more favorable microbiome metabolism. In hens, the better profile aligned with a more stable behavioral state, lower FCR and tryptophan metabolism being less shifted toward the kynurenine pathway. From a welfare perspective, these findings suggest that birds within the same group may not experience the environment equally. So social rank was not just a social label, but a biologically meaningful source of variation within the flock. And that's all my presentation. Thank you very much for your attention.